that we talked about from evolutionary theory and prenatal development into a nice little package. And I think that package, if we're successful, will provide a foundation to move forward. So when we're thinking about developmental disorders, we have a sense of what's going on. We may not know specifically what's happening, that's why we're taking the class, but you'll at least have a sense of the underlying mechanisms in some way, right? And so we're going to start with DNA. And I suspect you all know this, but DNA gets its name why? Sorry, you're whispering.
Is that the deoxyribonucleic acid?
Right. And it derives its name from the sugar. So in DNA, the sugar is deoxyribose, and in RNA, it's ribose. Fair enough? Now, the basic unit of DNA is the nucleotide, as I suspect most of you are aware. So the nucleotide is composed of a nitrogenous base, a sugar, and a phosphate. Now this is going to be important. Computer's down. And this is important when you start to look at our double helix. Now, of course, in the cell, if you're not preparing for division, then you're not going to have a bunched up double helix where you can perfectly articulate it, right? But let's look at it in this stylized version. When you see the double helix, if you take this strand here, the big line, that is your sugar and your phosphate. That's the backbone, right? Here, this is your nitrogenous base. These are your nitrogenous bases. And of course, they bond with our second strand through a hydrogen bond, right? We have binding there. And this is going to become very important for our topic as we move along. So if you're wondering why does this matter for this class, it will. And so we know a couple things. We know that we have, when we're thinking of nitrogenous bases, we have purines and we have pyrimidines. We all know that's true, right? Purines bind pyrimidines. Purines don't bind purines, and pyrimidines don't bind pyrimidines. Now, there are rare occasions where it might happen. So you might have, for some crazy reason, an adenosine binding adenosine, but we have elaborate internal mechanisms to correct for that, because that would be a disaster on par with Pearl Harbor or something, if that was allowed to happen. And so, when you're thinking about purines and pyrimidines, it'll help you to memorize these if you think of the purines as, and you say the simple phrase, pure as silver, or you could say pure silver if you want, and the elemental symbol for silver is Ag. So if we're transferring that over to nitrogenous bases, what might be the purines? There we go. And for pyrimidines, we can say cut the pie, P-Y instead of P-I-E, cut, C-U-T. So that gives us, wait, so louder?
No.
There we go. And uracil is RNA. The only difference between thymine and uracil is a methyl group. That's it. But when you see them, especially if you're starting to look at it, if you see that methyl group, you know you're dealing with uracil. It's not necessarily super important for our context, but you should know it. And so if you can think about cutting the pie and pure silver, then you should be able to keep them sufficiently straight. Now, any questions about that so far? We're going to come back to it. It'll be important, but we're going to take a break from it for just a second. And we're going to talk about meiosis. And we talked about it on Friday, and I don't feel like I need to spend a lot of time dealing with it right now. But there's something that I feel like we ought to kind of have clear in our heads. And so tell me if these things seem untrue. But generally speaking, we know that if say mom gives us this chromosome and then dad contributes this one. So our chromosomes look like this, right? When we get the X shape, we're dealing with replication, right? We all know that's true. If you didn't, it is. And so, by the way, so we can back up if we're going to thinking about definitions. What you have here is a single chromosome, right? Now, where it gets confusing is once you have replication and you have this kind of thing, and you have binding with the centromere here, now this is also called a chromosome. And what these end up being called, these two what would be different chromosomes are called sister chromatids, right? And so don't get super wrapped around the axle, at least for this class, about those things, but I know sometimes if you haven't been exposed to it and all of a sudden you're seeing chromosome thrown out in different places, but the pictures there don't look the same, it can be off-putting. But let's say this has happened, right? What you can have is a concept called crossing over between sister chromatids, and you can also have crossing over between these, right? And so if you have crossing over of these chromosomes, the arms, then that is one way that you're getting the new genetic combination that is you from mom and dad. Does that make sense? Now, those of you that are studying that sort of thing will know, and if you're not, you should expect that it's much more complex than that. That's kind of a very basic gloss. But it's important to know that something's happening and it's not magic, right? Any questions about this stuff? No? Okay. Okay, so if you have your notes, and you look, you'll see we're going to move into relatedness, genetic relatedness, and you'll see, if you don't have them, just when you see them, you'll see this. You'll see that there's this picture of siblings and a small chimpanzee, subadult chimpanzee, and the reason why that picture is there is because I want to take some time, a very limited amount of time, to clear up the idea of being genetically related. And so, two things will emerge. One, we'll talk about siblings, whether they're dizygotic twins or they're just siblings at a different time period. So maybe they're a year and a half apart or whatever it is. And you'll see that we say these siblings share 50% of their genetic inheritance, right? But then we'll talk about the human genome. And we'll say all humans, no matter where they are, share 99.9% of their DNA, their genetic stuff, their genome, right? So both those things are true, and sometimes that can seem confusing. And what's going on is we're talking about apples and oranges figuratively, right? So when we're talking about 50%, we're talking about contributions from mom and dad. So siblings are 50% similar because they're getting, this combination from mom and dad. When we're talking about us being related to other animals, we're talking about on the full genome level, right? Okay. And so we're 85% related to mice. I think 80% for cows, which always confused me.
50% to a banana.
That's that's why we don't get along, bananas and I. It's like Thanksgiving all the time, family fights. And you can do that for most things, right? You can go down and see what our similarity is through genome mapping. Now, you'll see that there are 3 chromosomes on your sheet. And so 2 things I want you to get from that. One, you can look at the similarity for this particular chromosome between our chromosome and the homologue for chimpanzee. And you'll see visually it looks identical for all intents and purposes. And that's true for a lot of things where there are slight variations. And so naturally you might wonder, well, why are we so different then? Certainly not all of the genes are identical, but even for the ones that are, we have to reconcile differential expression. So not all genes express the same way between species, right? So we might have the exact same gene, same place on the same chromosome or a homolog as X animal, but maybe we really express it and they weakly express it, and you're going to get a different result, right? Now, we're going to bring this together, and hopefully this will help make the evolutionary theory lectures make sense. And we're going to talk about single nucleotide polymorphism. So we said at the beginning that a nucleotide, which is the basic unit of the DNA molecule, is made up of a nitrogenous base, which it might be, a sugar, and a phosphate, right? And so when we talk about a single nucleotide polymorphism, what we're saying is that that nitrogenous base was switched out. So someone didn't do the switching, but for whatever reason, instead of getting an A, you got a G or a C or whatever it is, right? And that can be a problem. Often it isn't, and here's why. Let me draw our guy here. So it's doing this thing, and somewhere here, we have our centomere. So we're getting ready to replicate. And let's say we have these genes here. Just pretend they're identical, right? So it's going to move apart. Now, in between, and you'll see pictures on the internet if you want to see where we've had dye taken up and you can really visualize the genes. Even though the chromosome is just one big strand of DNA, right? It's all compressed. Not everything in that DNA molecule is a gene, right? So you can imagine a rope, and then you have a piece of tape here, and a piece of tape here, and a piece of tape here, and a piece of tape here. And that's how you can think about it. Now, when we think about single nucleotide polymorphisms, if we say on average there are 10 million SNPs in the human genome, just the law of probability tells us that the majority of those SNPs are happening in what we used to call junk DNA. You'll hear people that learned when I learned still call it junk DNA. We of course know it's not junk now. It's hard to change terminology once you have it kind of buried in your head. So at one time it was thought that the DNA sequences here really didn't do anything, but what we now know is it does quite a bit, and one of the things it can help trigger expression, gene expression. So that's for a different class. But why this is important is because if we're thinking about natural and sexual selection, especially natural selection, we said that we have random mutation and non-random selection, right? So the random mutation could be a SNP, a single nucleotide polymorphism, and let's say it happens to land on a gene. There might be a problem. Let's say that it lands on the gene that helps your feet develop, and you are a pronghorn sheep, and for some reason your back right foot is clubbed out. So now, because you live in the Pleistocene, you can't get away from the cheetahs that used to roam on the Great Plains here. You will be selected against, right? That mutation. So the cheetah is going to run fast regardless of what happens to you, whether you have that SNP or not. You don't have it, but having it, if it causes such a disadvantage, well, you'll die. You won't get to reproduce. You won't pass that along, and there we have it. Now, if, however, and we know that expression, usually this is multifactorial, but for simplicity's sake, let's say you have a SNP that contributes to you going faster than other pronghorns. Well, the cheetah is still going the same rate, right? Mostly. you have this polymorphism. You can now run faster. So not only can you run faster, but you can get away from the cheetah and you can reproduce and have lots of little pronghorn babies and all of that sort of thing. So that's all well and good, but eventually, right? Eventually, we're going to have that same thing with the cheetah. So the cheetahs that can run just a little bit faster because they have a polymorphism that allows for it are going to catch more pronghorns. That means they're going to be able to feed themselves and their offspring, and they're going to do better. So we have this constant arms race, and a lot of it's based on what's happening at the nucleotide level. Now, you can have larger polymorphisms. We know that's true. But you'll see that SNPs are probably the most common. That brings us, of course, into the red queen hypothesis. Now, you'll see in your notes that this is pulled from, and a number of people use this, is pulled from Lewis Carroll's Through the Looking Glass, Alice in Wonderland, which I've told the other classes, but I'm going to tell everybody because I believe it's my job to shout it from the rooftops. The Queen of Hearts and the Red Queen are different people. The last movie, they put them together into like some crazy amalgamation. They're not the same person. The Queen of Hearts is a card, and the Red Queen is a chess piece. It's very upsetting. But at any rate, it comes from this idea. So if you happen to read it, Alice and the Red Queen are running this race, but they're not getting anywhere. They're not going anywhere. It's almost as if they're running in place, right? And Alice very calmly remarks to the Red Queen, in my country, when we run a race, generally we get somewhere. And the Red Queen responds by saying, well, that seems like a slow sort of country. Here, if you want to stay in the same place, you run, but if you want to get somewhere, you have to run twice as fast. And so that's been used as an analogy for this arms race. So, the contribution of sexual selection to this arms race. So we have here these polymorphisms that may give it an advantage, may give a disadvantage, may do nothing at all. But this is coming, a lot of this, through sexual selection, right? We're getting some stuff from mom, some stuff from dad. If you could reproduce asexually, then your offspring would be identical to you. So while the other organisms are all adjusting to be better and faster at what they want to do, you would be exactly the same way. It puts me in mind of this apocryphal story of Poland in World War II, where Hitler comes into Poland with his tanks and stuff, but Poland had been isolated, so they didn't know that they needed to kind of change their armament, and so they met the first Panzer unit on horseback. They had knights with lances because that was their kind of elite unit. Of course, knights and lances don't do anything to tanks. And so this is the same thing. We're the viruses, you might imagine, or bacterium might be continually evolving, and they have much shorter generations, so they can do it much more rapidly. And they're coming at you with a Panzer unit, and you're, throwing up your shield and heading off to the Ren Fair. That makes sense? So sexual reproduction helps us run twice as fast, because not only are we getting the good stuff that worked with us mostly, but we're getting potentially good stuff from this other person. Either way, it's getting mixed up, so we're not staying the same way. You can also think about it as a code breaking, right? If you keep the same code and you're constantly talking in code, eventually someone's going to break it. If, however, you continually revise your code, whatever it is, it's going to be much harder to break that. That all make sense? It's very exciting. Very exciting stuff. Okay, I think that's all I want for that. On this page, so I have these two cartoons. I suspect you're familiar with these. This, I suspect you've been seeing, you've seen since middle school, right? Kind of what happened in the fallopian tube. We're not going to spend any time on this unless you have a specific question, just because I think you guys already know it. Now, when you're looking here, something that I think is important that we won't spend too much time discussing, but it's important to think about, is when you get to the blastocyst phase, that's when most often we obtain embryonic stem cells, right? And as you all know, there's a big controversy about this, and I don't really care one way or the other where people fall, but I think whatever an argument is, it ought to be based in physiology, at least in this case. And so blastocyst, of course, is just a collection of cells. It's not a creature at all. The vast majority of blastocysts are spontaneously aborted. They never implant, or they do implant and then they're spontaneously aborted. It's actually much more rare to have a successful implant into the uterine lining. And so this happens within the first 2 weeks, right? So you might not even know. You could have been pregnant 10 times and not know it, but it was just a blastocyst that went by. Not here, though, not at Augustana. Like, do that sort of thing. And so you may come on the side and say, you know what though, there's potential there, and the potential is what's important, and that's why I am over here. Or you may say it's nothing. It's like pulling out cells before neurogenesis, and they're not doing anything. You can make them do whatever you want, and so it really doesn't matter whichever side you fall on. I really don't care. But you should base it in what's actually going on, and not some kind of nebulous idea of what's going on. And so hopefully you'll be able to work on your arguments, whichever they are, throughout the course of the semester. For the stages, you'll notice that I provided this little chart here. This is from your book. It doesn't look exactly like it, but it is from there, and that should help you kind of condense some of the information. Now, those of you on the quiz that had trouble with the mesoderm question, you'll see in the textbook where it talks about the three general layers that we have within the embryonic stage, so 2 weeks to roughly 8 weeks. And so, of course, the ectoderm, which is the outer layer, and you can make whatever Ghostbusters joke you want to mentally at this point. Ectoplasm, right? Quit being so boring. And so from the ectoderm we get skin, hair, teeth, sense organs, CNS, brain, spinal cord. The inner layer is called the endoderm, and from it we get digestive system, liver, pancreas, and respiratory system. And then wedged in between those we have the mesoderm, right? And so we have bones, muscles, blood, your circulatory system, all of these great things. And during this period, we also have rapid neurogenesis. We're going to talk a little bit about that. It's one of the areas I tend to rant on because I love it so much, and I think it's quite amazing. But here we start to have not only the creation of neurons, because it's not necessarily true that they're going to become neurons. They could become glia, right? And of course, neurons don't divide. So this whole stage where they're either becoming neurons or they're becoming something else, or they're kind of being shuffled back in is quite amazing. But what you would want to know is that it's during this phase that if you have the appropriate equipment, you could detect weak EEG readings. Very weak, but you could do it. The fetal stage, your book has it listed at 8 weeks. That's a rough estimate because it's not like a graduation ceremony. Well, it is kind of, but it's not you get to 8 weeks and you go all of a sudden you were an embryo and now you're a fetus. You're a fetus when your major organs have differentiated. So that's the point. Now, no one's walking around with the equipment to judge that on a daily basis. So at 8 weeks, you could happily say, yeah, I have a fetus now. I don't know if people do that, but you could. And so we have an increase in neural and somatic connectivity and complexity. So what that means is within the body and the brain, not only are these things growing and developing, they're starting to connect with each other. So we're starting to have functioning systems, which is, of course, very important. We see the beginnings of sleep-wake cycle. This is not the same thing as circadian rhythm. That takes a while. That's after birth that starts to happen. Wow. And it makes sense why that is, because you have to have an operating SCN and you need photons to do all of it. Yes. So it's not circadian rhythm, but you can detect sleep-wake cycles. When we say detecting sleep-wake cycles, that's via EEG, not, so we're looking at brain waves and saying, okay, this is a sleep stage, this is a wakeful stage, which is easy to do, easy enough to do. It's not kind of looking at, ultrasounds going, oh, eyes are closed or something silly like that. And this, of course, is where you have gender differences starting to occur. Now, it's not as simple as reproductive organs. The majority of what's going on is happening in the brain. So when you have these hormonal cascades, it's not only is it helping the growth of genital or reproductive organs, it's also changing how the brain is wiring. Now, of course, there aren't a ton of differences, but there are some differences. We have sexual dimorphism. It's maybe not to the same degree that we see in other animals, but it is there to some degree. And so you'll see when we get into later chapters, so I think adolescence is where we first encounter it as a chapter, that we'll talk about things like being cis versus trans or whatever it might be, or different sexualities, and we'll talk about that in the sense of within the framework of neuroscience and what has gone on, and we'll talk about why choice really isn't kind of in there. Teratogens is the last thing that we'll touch on just basically. So I've provided this nice chart. I'm not going to spend a ton of time going through each thing, and the reason is because when we get into birth and the newborn and as we go through, we're going to deal with a lot of these stages individually. Even though things are happening in the prenatal period, we don't usually see them manifest until we're out of the womb, right? And so we will be talking about them. But you can think of any insult that causes fetal death or affects the fetus in some significant way would be a teratogen. So the last thing I'll say is your textbook, which I don't agree with, says, it gets the part, what's the role of the father? And it says, maybe this, maybe nothing, I don't know. And here's the thing. Certainly we can all say that secondhand smoke is a problem. There's no way to do secondhand alcohol that I know of, unless you're holding the funnel in the person. I don't. So there's no real way to do that. But one of the things that I think is very important, I'm going to talk a lot about this semester, is stress. And so that could be the father. That could just be an environmental situation. But what we see is that stress is extremely potent. And you're going to hear me talk a lot about this guy named Robert Sapolsky, who I'm a big fan of. And he's one of the first researchers to kind of uncover what's going on in the stress response. And what we see is that when the HPA axis is activated, we end up with cortisol flood. In people that live in chronic toxic stressful situations, you see this spike of cortisol instead of coming back down to baseline, which is healthy. So there's no such thing as good stress and bad stress. If it's good, it means you spike and then you recover. If it's bad, so it's prolonged, you go up, you never come down to base, and you kind of do this, right? And so you're leveling off way above baseline, which means that you can spike, you can have a cortisol spike with smaller insult, a smaller noxious insult, and you constantly have this bathing of some level of cortisol, which can lead to apoptosis and all this other crazy stuff. And we'll talk a lot about that. And certainly arguing parents or no support network can do that. But we are right at the hour.